Hello, good morning, and welcome to this tutorial. So, we're going to cover briefly signal transduction, and we'll go over a few questions that came in test one and test two that pertain to signal transduction. So, let's get right into it. So, signal transduction, if you're trying to gun it or trying to understand it last minute, basically has a few components that you have to take note of. Number one, that is the ligand that binds to receptors. Okay, then the receptors themselves, then you have second messengers, and then lastly you have what those second messengers do. So that is basically what signal transduction is in a nutshell. So if you try to define it, signal transduction is basically uh, coupling of an extracellular stimuli with an intracellular response. That is basically what signal transduction is. And then those components work together, just like I mentioned before, to ensure that the cells communicating. So signal transduction is also called cell signaling, by the way. It's how the body communicates to the cells to do particular things at a particular time. Okay, so to get right into it, like I said, you need a ligand, you need a particular ligand that will bind to a receptor. Let's look at that as a receptor. And this receptor is basically on the membrane. We have some intracellular receptors, of course, but for this example, let's use the one that is on the membrane. And this one, for for signal transduction purposes, the topic that we're covering, the best example we can use, rather the best in the coverage, basically, you have what are known as G-coupled receptors. So you have a receptor, this is a receptor, and they say that's a ligand. And then you say this is coupled to a G protein. So this is a transmembrane protein. We know what transmembrane proteins are by now. Then it's coupled to a G protein complex that has alpha, beta, and gamma subunits. Okay, that is G coupled receptor system. So this arrangement allows that once you have the ligand binding to this receptor, it's going to activate this G protein coupled this G protein that is coupled to this receptor, and then the alpha subunit is going to be released, and then it's going to bind to adenyl cyclase, which is on the inner membrane. Okay, on the inner, not really the inner membrane, but on the inner part of the bilayer. So we have what is known as adenyl cyclase here. Adenyl cyclase. Okay, so what is the purpose of this adenyl cyclase? Adenyl cyclase, basically, from the name, it's going to cause formation of CAMP. Okay, so adenyl cyclase basically gets AMP and cyclizes it, forming cyclic AMP. Alright, so the alpha subunit goes to adenyl cyclase, then activates it to form cyclic AMP from AMP. So once the cyclic AMP is formed, it now has a very important role. This cyclic AMP is going to go and activate what is known as a protein kinase. Okay, so a protein kinase is going to be activated by cyclic AMP into the active form, which is activated protein kinase. Let's use that for now. So this activated protein kinase, we know that kinase enzymes basically are involved in transfer of phosphates, and phosphates are basically the way in which it's more like an energy currency of the cells, basically. That's why we have adenosine triphosphate, monophosphate, diphosphate. So the phosphate parts are the ones that the phosphate bonds themselves are the ones that are responsible for transferring energy. So if you're transferring a phosphate, you're basically transferring energy. All right. So when you're transferring energy, you use a kinase enzyme. So what this kinase enzyme can do, it can activate a protein. This protein can be an enzyme, because remember that enzymes are also proteins. So what we are saying is that we can activate another protein into an active protein by the action of this protein kinase. So that is basically what the G protein coupled receptor system is all about. So this protein, sometimes it cannot be a protein. It can go to a membrane associated receptor that activates transcription of a particular protein, and that is also one mechanism in which they act. So this is the mechanism in which cyclic AMP, for example, acts through the G coupled receptor system. So what I've just explained here is just a brief overview of G coupled receptors, and in case you're asked about CAMP, this is basically what you need to explain. Okay, that is, and then a perfect example, if you are being asked for CMP exactly, the perfect example is its role in glycogen metabolism and um, what is this? Glycogenolysis, yes. Glycogen metabolism in terms of glycogenesis and glycogenolysis. It's going to inhibit glycogen synthase while it's going to activate um, this enzyme, glycogen phosphorylase. So glycogen synthase is responsible for producing, for forming the glycogen uh, chain. What was the best way to put this? The glycogen branching chain. The this entire glycogen um, molecule. Is it right to call it a molecule? I'm looking for a word to best describe glycogen, but yes, it's responsible for synthesizing glycogen by adding those 1,4 bonds uh, there, adding glucose in those 1,4 pattern bonds. While glycogen phosphorylase does the opposite, it breaks down glycogen into glucose 1 phosphate monomers. So these enzymes act differently, but they are all activated by the action of cyclic AMP. But the way they act for glycogen synthase, if you phosphorylate it, it's going to be deactivated, it's going to be inhibited. Then when you dephosphorylate it, when you remove the phosphate, you're going to have its action being restored. But for glycogen phosphorylase, this one, once you phosphorylate it, you add a phosphate to it, it's going to be activated. But when you remove the phosphate, when you dephosphorylate it, it's going to be inhibited. So please get the difference between these two. And that's the action of cyclic AMP. All right. So with that said, we're going to go over a few questions that okay, maybe before we go there, there are also cyclic AMP is classified as a second messenger. So second messengers, uh, there's a number of them that we can take note of, CAMP being one of them. We also have um, diacylglycerol, DAG. Okay, I'm gonna write that properly. So we have diacylglycerol. We also have inositol triphosphate. We also have phosphatidyl uh, inositol diphosphate, 4,5 diphosphate or bisphosphate. All right, so and calcium as well. So these are basically the second messengers that you have to take note of, basically how they act in the. So, yeah, so there are questions that came and it required you to have an idea of what this is. So we're going to go over them and then we basically put a lead on what they really are. All right, so let me just scan the questions and see which ones required. So there's question five. I'm sure this is from test three. Question five from test three, and also test question one. Okay, so we're going to start from question one and then go to question five, and then there is also question the last. Then if the last question from test two, and then we'll wrap it up. Okay, so the first question from test 3 says that explain how second messenger systems such as cyclic AMP amplify the signal of peptide hormones. What is signal amplification and what is its importance in hormonal responses? Okay, so second messenger system, as I don't know if I defined it earlier, but second messenger system is basically an from this definition that we have here, is a cellular signaling mechanism that transmits signals from the outside of a cell to its interior, leading to a specific cellular response. You can put these words however you want them, but that's the basic idea of what second messenger system is. It's a way in which cells translate um, an outside signal to an intercellular signal and promote, or rather, yes, yes, stimulate an intracellular response. So, uh, the role of cyclic AMP, basically, like we said from that diagram, I wanted us to start with this question because I'm just from saying this. The role of cyclic AMP is to activate a protein kinase, an enzyme that will phosphorylate another enzyme or another protein, because it may not just be an enzyme but just another protein, thereby activating or inhibiting it. And we give examples of uh, which enzymes are activated or inhibited, and um, yeah, by this activation of uh, the phosphorylase, the kinase rather, protein kinase. Okay, so this is a brief idea of how the second messenger system is. So as you are describing, this question requires first, if you check the backing key, this requires that you describe what a second messenger system is. So this is what we are doing in this first part, and then in the second part we are now going to what signal uh, amplification is. So signal amplification is basically from the two words. Amplifying a signal is basically making the signal stronger. So um, how do you make a signal stronger in cell terms? Okay, so and how is that important and how does that actually happen? So for signal amplification, one way in which the body has designed signal amplification to work with regard to second messenger system, you're going to find that as we checked the ligand, in this case it can be a neurotransmitter. Another example, a neurotransmitter like epinephrine. Okay, so the essay requires that you also give an example. So epinephrine is an example neurotransmitter. One molecule of a neurotransmitter or epinephrine, as it binds to the receptor, is going to activate about 100 of them. So hundreds of G protein receptors or G protein coupled receptors, um, rather G proteins that are coupled to the receptor. Let me just retrace my steps there. So you have one receptor, and I always draw receptors like this for some reason. So one receptor, it is coupled to hundreds of G proteins, those ones that have alpha subunits, gamma, and beta subunits. Those ones, those G protein complexes. There are hundreds of them coupled to this receptor. So once epinephrine binds, hundreds of these are going to be released. Okay, so once they are released, ultimately we know what that the alpha subunits of these hundreds of G proteins are going to activate another hundreds of adenylocyclases. Okay, so hundreds of adenylocyclases are going to produce further a lot more activation of protein kinases, because adenylocyclase produces cyclic AMP. So if you produce hundreds of molecules of cyclic AMP, then hundreds of uh, protein kinases can also be activated in that manner. So you're having more like a cascade. One molecule activates hundreds, then those will activate another hundred. So that is like a cascade. You're amplifying the signal at each and every step. You're making more and more responses from the initial response that you have. So that is basically what signal amplification is. Okay, so what is its importance? The final part of this um, question, of answering this question. So the importance, number one, it prevents signal loss. Prevents signal loss. So imagine if it was a stepwise. We have about three steps in G coupled receptor systems. One, home ligand binding to the extracellular receptor, and then you have a G coupled protein that is attached to that, alpha subunit, beta subunit, and a gamma, and then you have adenyl cyclase, and then you have a protein kinase. Okay, so imagine if just one, uh, this was happening with just one molecule is released on each step. Now imagine something happens to that very one molecule. Let's say this alpha subunit, when it's coming to adenine cyclase, it's maybe inhibited by something, or maybe just something, because basically you don't have one process happening in the cell. So you might find that there is an interference by something else that is happening, another metabolic process that just inhibits the this same process of the alpha subunit moving to that, because there are proteins and a lot can happen in the cell because there's a lot of metabolic activity happening in the cell. So if you cut that, that means this signal is cut completely. This chain is cut completely. But if you have hundreds of them doing the same thing, that means even if you cut one off from activating the adenine cyclase, because you are releasing hundreds of these G proteins. That means obviously one of them, at least, not even just one, at least a good number of them is going to bind to adenine cyclase, activating it, and the rest of the pathway still goes. So that is how you prevent signal loss. This is how you explain this point. Then energy conservation. Energy conservation comes in the sense that you are not going to utilize a lot of hormone, okay, a lot of hormones uh, uh, for you to activate a particular cellular response. Remember, the body is big, and uh, the way hormones act, they're in small amounts, small concentrations. So those small concentrations can be translated into bigger responses just by uh, the signal amplification that we are talking about. So it conserves energy. You don't have to produce a lot of hormones for you to have a desired response. Just a small concentration is enough. Then efficient signal propagation, it still goes back to the point that we said that it's a lossless kind of transmission where uh, no information is lost, and you're going to have that efficiency, because efficiency is basically defined by how much of the target do you reach. So if you are reaching the target every time regardless, that means you have high efficiency levels. So those efficiency levels are brought about due to having hundreds of these G proteins that can be activated, and then making sure that at least a good number of them uh, are still going to be activated, as a good number of adenine cyclases, a good number of protein kinases are still going to be activated, even if some of them were to be cut off. So that's good efficiency. Efficiency measured on how well you're able to reach your target. And then finally, talk about sensitivity to lower hormone concentration. Because of the energy conservation system, that means even low hormone concentrations are able to be detected, because just one molecule of a hormone binding to the receptor means it's going to produce that cellular response. Obvious. Right. So that is how you can tackle this question in case they choose to bring you signal amplification again. We can move to question number 5, and then we'll go to that other thing. All right, so question number 5 says describe the role of intracellular calcium as a second messenger in the mechanism of action of hormones like oxytocin and vasopressin. All right, so oxytocin and vasopressin, very, very interesting hormones, both of them coming from the posterior pituitary, and it's known that they will both act via CA2 as a second messenger. All right, so hormones like oxytocin and vasopressin do act through calcium as a second messenger, and basically the way it happens, it's a cascade. It's another uh, coupled system. Okay, it's another coupled system that I'll try to explain uh, briefly down here, and then we'll go back to the question. Okay, this is taking time. Isn't there a way to just clear all the annotations? Um, 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 can't find a way to clear annotations from. Alright, there we go. Okay, that's just hiding and not clearing. Anyway, let me just try to to use this space. Can actually. All right. So as I was saying, there is another system that works with calcium. So it all starts the same. A ligand will bind to a receptor in the membrane. Okay, and this receptor is also bound to. Okay, so this receptor is bound to another molecule. Let's call this PIP3. That's phosphatidylinositol 4,5. It's bisphosphate, so it's PIP2. So this PIP2 is going to dissociate into once it's um, once it's activated, it's going to just dissociate into two molecules. It's going to break down into two molecules. That is IP3, inositol triphosphate, and the other one is DAG, diacylglycerol. Okay, and these the way they act, diacylglycerol is going to cause activation of um, a phosphor. Let me just try to check if that is the right. So it's rather a protein kinase, not a phosphokinase. Okay, so protein kinase C. Then inositol triphosphate is going to cause release or mobilization of Ca2 plus from uh, the plasmic reticulum. Usually, those are the calcium stores in the cell. So protein kinase, for it to act, it depends on calcium. So that's why it's called protein kinase C. So this calcium, once it's liberated by inositol triphosphate, it's now going to be uh, required by the protein kinase C, and then the protein kinase C can now work because it requires this calcium for it to work. So this is how basically that happens. So you're going to have binding of the ligand to this receptor, and then that receptor activates um, protein or rather phosphatidylinositol 2 or not 2 4,5 bisphosphate, and that coupled with other other factors like growth factors, for example, are going to cause lysis uh, of this molecule PIP2. It's going to break down, and it's called hydrolysis because water is involved. So once it breaks down into two components. These are inositol triphosphate and diacylglycerol. So these two work hand in hand. Diacylglycerol works by activating this protein kinase, while uh, this inositol triphosphate is going to cause release of calcium because the protein kinase C, for it to be fully active, it requires calcium, more like a cofactor or something. So then that is how this uh, system works. So the question, going back to the question 5, the role of intracellular calcium as a second messenger and mechanisms of hormones like oxytocin. So this is basically how you can structure your essay. So these are just pointers on how you can structure your essay basically. Talk about hormones like oxytocin relying on Ca2 plus as a second messenger. Then binding of either of these hormones receptor activates the phosphatidylinositol 4,5 bisphosphate PIP2 on the inner leaflet of the bilayer. So the inner leaflet of the bilayer has PIP2. So bilayer, and then just the way adenine cyclase is, we have PIP2 there. So that PIP2 is going to be activated, just like I said earlier. Then growth hormones and other factors stimulate hydrolysis of this PIP2 by phospholipase C, and diacylglycerol and IP3 are produced. So IP3 are going to be produced, IP3 and diacylglycerol. I'm just repeating here. Are going to be produced, and then actions of Ca2 are mediated by mostly by calmodulin. Okay, so for the for the Ca2 to act as an intercellular signaling molecule, it requires that it binds to calmodulin most of the times. But another way in which it can act is via protein kinase C, just like I mentioned, protein kinase C, and this one requires both DAG and IP3 to be involved, like I said. IP3 for releasing calcium, and calcium can either go to bind to calmodulin or bind to protein kinase C, which was activated by diacylglycerol area. Okay, so with that mentioned, the cellular responses now for this question is just 5 minutes, so I don't think you need to go in any more detail than this. But yes, cellular responses include exocytosis, for example, contraction of muscle. So in relation to oxytocin, you relate contraction to oxytocin, like contraction of the uterine walls, for example, during parturition. You you can relate that to oxytocin, and yeah, contraction, apoptosis, cellular programmed cell death, activation of immune cells as well depends on calcium as a second messenger. So that is basically it also on question number 5. Once you just go over these steps that I mentioned, and yeah, you can score some good marks on that essay as well. All right. Okay, that is it for this paper. The other question was from test 1, from test 2 rather. I've forgotten the question number, but let me just find it. Oh, this is the last question. All right. The role of cyclic AMP in glycogen and glycogenesis. This was supposed to say glycogenesis. This was supposed to say glycogenesis and glycogenolysis. Okay, so um, this one I already said this was just 5 marks. So all you have to explain is activation of glycogenolysis, phosphorylation of glycogen phosphorylase, which is activating it. Like I said earlier, I already mentioned an inhibition of glycogenesis um, by uh, phosphorylation of glycogen synthase. So you inhibit glycogenesis by phosphorylating glycogen synthase. Remember that this hormone, once you phosphorylate it, rather this enzyme, once you phosphorylate it, is going to be deactivated. So that's basically what you have to mention here. So these other points, this is just explaining exactly what I was from mentioning when I was talking about activation of glycogen synthase, or rather inhibition of glycogen synthase and activation of glycogen phosphorylase. With that being said, that is the end of this short tutorial on signal transduction with the questions that came in test 1 and test 2.